All right, I'm gonna get going here. So thank you guys for coming. Thanks, Vito. Yeah, so our quantitative systems biology seminar series is happy to have Dr. Natalie Stanley here with us from University of North Carolina in Chapel Hill. She's an assistant professor in computer science and computational medicine. She got her undergraduate degree in mathematics from Dickinson College, and then a PhD in computational medicine, computational biology. That's it. And then went on to Stanford for her postdoc with Nima Agyapour, where she started delving into immunology, her first foray into immunology. But the work that she had done on graph theory before and that work in immunology has led her into starting her own lab and developing her own work at UNC. And she's got some really cool tools that she's been developing and starting to apply them to some very interesting questions. She's just now getting into some neuroscience with gliobiology, and I'm very excited to hear what she has to say about the work that she's been doing. Thank you.
Thanks a lot, Darren, for the introduction, and thanks everybody here at Vanderbilt for having me today. I'm having a really amazing visit here. I've had really exciting conversations all day, so I really appreciate the opportunity to be here. So I'm really excited to tell you about the work that we've been doing in this computational immunology space. I feel this is a really fun field to work in because we have all of this data about very different types of immune cells, and we try to basically characterize these cells and then use signals that we can extract from them to say something about particular clinical phenotypes, or maybe even to come up with new treatment strategies. So today I'm going to tell you about basically how we take a bunch of profile data and turn that into some kind of nice signal or feature representation that we can use for downstream prediction tasks. So, this word featurizing, that's the first word of the title. You can just sort of think about this word as meaning, how do we take a bunch of cellular profiles and basically come up with a representation and a summary of the data so that we can use it for downstream prediction tasks. And my contact information is here, so feel free to reach out to me if you have any questions or anything after the talk. Okay, so the first thing that I want to do is I want to tell you a little bit about our lab and some of our collaborators that drive a lot of our questions. So the first picture is a picture of PhD students in the lab. And what's really nice about these students is that they all come from a lot of different backgrounds. So we have computational biology students, we have computer science students, and we have applied math student. And they all really bring unique perspectives to the lab and different ways in which we can, study these data and set up our problems. So broadly, as a computational immunology lab, our overarching goal is to identify particular components of the immune system that can really be modulated for therapeutic effects. And, this can be to design things like vaccines or maybe even more effective immunotherapies. So we're really interested in figuring out what's the best way, for example, that we can get a T cell to behave well, or what kind of cells does a person need to have in order for them to respond favorably to a treatment or to be protected against a disease. So a lot of the work that I'll be talking about today is with these collaborators at UNC. So in this talk, I'll be mostly focusing on work that I've done at UNC in collaboration with these people. So just to briefly explain sort of what they're motivated by. So Justin Milner is a T cell immunologist, and he works on designing effective immunotherapies. This is Junior, who helps with a lot of the machine learning work. Todd helps with neurodegeneration. Tony is a pediatrician that focuses on women's health and sexually transmitted infections. And Nilu works on HIV and HIV vaccines. So I'm going to be highlighting work from many of these collaborations today. Okay, so the first thing that I want to do is basically define what I mean when I talk about the immune system. So broadly, you can think about the immune system as a complex network of cell types and signaling pathways that synergistically work together to protect an organism from things that are foreign. And what's nice about the immune system is that it has this, nice hierarchical structure in which a primitive cell, like a stem cell, can give rise to many of these specialized immune cell types. And all of these immune cell types have very specific functions and basically roles in protecting organisms. So with modern technologies, some of which I'm going to be getting into in this talk, we can think about how do we take a sample of blood, for example, and distill that sample into its individual subunits, which are cells, and then basically determine what types of cells those are. So we can basically enumerate cells of all of these types within a sample. And nowadays, these technologies have even gotten so advanced that we can think not only of phenotyping cells or characterizing them, basically identifying them, saying what they are and describing them, but we can actually do things with imaging, for example, and make statements about how cells are organized with respect to each other and to other cells, such as tumor cells. So in this example I'm showing you here, this is a sample from a patient or a schematic of a breast cancer situation that you could have with tumor cells in blue, and the bright colored cells are immune cells. And it turns out the extent to which you have mixing between tumor cells in blue and these other immune cell types, this actually has prognostic significance. So in order to address a question like this, you would not only need to be able to differentiate between these different kinds of cell types, but you would also need to be able to encode information about like this mixing or what kind of cells were next to each other. So I'm going to sort of touch on all of these points in this talk. I wanted to give you a couple of examples of problems that I've worked on where the immune system really has characteristic changes that really drive particular diseases or health outcomes. So the first major area of focus that I had, especially as a postdoc, was in pregnancy and women's health. So there we looked at a couple of problems, such as what happens to the immune system throughout a woman in a woman while she's pregnant. And it turns out there's a lot of very characteristic changes in the frequencies and functions of particular immune cell types. So for example, how many granulocytes do you have? This will change as a function of your gestational time. And another project that we worked on was looking at patients with preeclampsia. So preeclampsia is basically a gestational hypertension disorder, and it's very serious for the mother if she has it. So looking at samples from patients with preeclampsia, we would like to be able to say at a very early time point whether or not a woman was likely to develop preeclampsia. And this can do things like inspire lifestyle interventions. And so it turns out that we can, just by taking blood samples and looking at immune profiles gleaned through the blood, we can actually make statements about who's likely to get preeclampsia. And then two other areas that I've worked on are understanding, different trajectories of surgical recovery. So why do people recover quickly from surgery and other people recover more slowly? There's differences in their immune responses after a big perturbation like surgery. And I'm going to be telling you a little bit about this story today, about how modulating inflammatory responses after surgery really does make a difference in recovery. And then finally, an area that I've become interested in is neurodegeneration and diseases of aging, where we want to study neurons, glia, pathologies, and immune cells, and how all of these things interact as a person ages or experiences neurodegeneration. Okay, so the main profile that we use to characterize all of the diverse cells in the immune system is called mass cytometry, or CyTOF. And this is a high throughput immune profiling technique which measures proteins in individual cells. So roughly how this works is that you first decide which proteins you want to measure in your cells. You find antibodies specific to those proteins, and you conjugate those to a heavy metal. And then you stain cells with these metal conjugated antibodies. And whenever these metal conjugated antibodies find their protein match, and they're passed through this fancy mass spec machine, you get some kind of a fluorescence. And so basically in a plot like this that comes out, basically all of the colors are representing different proteins. And so whenever you see a peak, this is a proxy for whether this protein was expressed. But for people like us, what we get out is this matrix of cells in the rows and features measured about these cells in the columns. And, for a single sample, the dimensions of this matrix are typically on the order of 200,000 cells and 45 to 50 different proteins. And while that doesn't sound like a lot, if you think about if you have hundreds of samples, so you multiply 100 samples by 200,000 cells, this quickly ends up being a lot of information. So our work comes in really in this phase, and that we want to basically, without bothering any human, right, to provide us with any input, we really want to be able to automate all steps of this analysis. Like the first part is figuring out these phenotypically coherent subpopulations of cells. And then we would like to basically define properties about these different cell populations that would allow us to predict things that we want to know clinically, or that we want to understand about some kind of experimental perturbation. So, when you start working with single cell data, you quickly realize that there's a bit of a learning curve because the data structure that's produced is so unique. So I wanted to sort of walk you through an example if you've never seen data like this before. So schematically, I'm illustrating here. You can imagine you have 4 different patient samples denoted by each vial of blood, and a different color is used to represent each sample moving forward. And then, so you can run CyTOF on each of these samples. And what this does is it produces a matrix for each samples of cells by protein markers or features that we measure about these cells. So you can also do this for genetic information like RNA-seq or ATAC-seq, or basically any kind of single cell modality follows this general data structure. So then ultimately what we would like to do is, now we're stuck with these multiple matrices of data, right? In contrast to, typical problems in data science where you just have one matrix of data and a vector of outcomes, now we have 4 matrices of data in this little example. And within each matrix, there's all kinds of heterogeneity going on, right? Because every row of these matrices are instances of a broader cell type. So they're sort of like bins of information here that need to be properly assimilated so that we can best summarize and explain the data. But ultimately, where we're headed is that we want to be able to summarize the data in a way that we could accurately predict, something about the patients. And all of this, again, we want to do algorithmically in an automated way, so that a human doesn't need to spend time sort of sorting these cells by hand and trying to make sense of the data in a qualitative way. So it turns out that the people in machine learning have been studying these kinds of data for a long time. And looking at point cloud classification. So if you haven't heard the term point cloud, this is just a bunch of, for example, a bunch of data points in some d-dimensional space, where in this example, I'm showing you points in 3-dimensional space, where each one of these little nuances of, let's say, an airplane is sort of an instance of a point cloud of a particular type. And on the other hand, we could have these point clouds that look more like chairs. And it turns out in machine learning, they actually have been working for a long time on how do you classify point clouds by type, right? So, this problem is really similar to our problem because we don't have little toy problems with chairs and airplanes. But if you think about every sample profiled with a single cell technology, they're effectively one of these chairs and one of these airplanes. And instead of living in 3D, they live in 45 to 50 dimensions, right? And so now you can think about every point here as a cell. So you can imagine each of these little images is representing a sample, and every point is representing a cell. And with this in mind, there's a lot of machine learning tools and deep learning tools where now you can just really throw in the data and get an accurate classification or diagnostic of the samples. So I am going to be telling you a little bit how to do something like that, but before we get there, I'm sort of going to talk about how we first went to automate the classical manual way of analyzing these data. And so a lot of the algorithmic innovations that I've made have really been inspired by a particular data set that I worked on as a postdoc, where we had to really do things like this at scale. So we had over 6 million cells in this particular data set because we'd profiled 2 different types of patients longitudinally after surgery. So we had, a pre-surgery time point and then several time points after. Some of these patients received a steroid called methylprednisolone after surgery, and the other were just controls. And then we looked at differences in their immune system, but this was so many cells. And if we wanted to predict, for example, different post-operative outcomes and actually throw in data from each of these samples into a model, we really had to do something a little bit smart with the data. So some of the problems that this inspired would be, how do we actually encode the immunological landscape with a feature representation, right? Similar to how I showed you with the point clouds, how could we represent every one of those point clouds as a single vector or a mathematically abstract representation that would allow us to do some kind of prediction? How do we deal with the large number of cells? Can we actually throw some away without losing any information? And how do we downsample the data such that we don't lose any of the important signals in the data? And then how do we really explain to our collaborators which cells are prototypical in the data set or associated or highly indicative of some particular outcome of interest? And so, just to reiterate, whether you have CyTOF from blood, which is what I've been describing, or from a tissue, and then you end up with your cell by feature matrix, what we want to do is take that matrix and compress it into a single vector. So then when we give that vector to a model, we can then, and we assume this is some kind of machine learning model that predicts an outcome, we want this vector to be such that we can then train a model to predict this outcome. And so this is really sort of the main theme of my work, and doing this in a way that's scalable and practical and can fit in a GPU and can, ideally be done on a laptop, but we ought to be a little bit realistic. Okay, so the original solution to this problem is with manual gating, right? Where you would run your single cell experiment. Typically this would be something like flow cytometry, and then you would take all of your cells and you would sequentially project them in 2 dimensions based on characteristic combinations of markers. So the first couple of steps, you're getting rid of dead cells, and then you do your first projection on CD45 and CD66, where you get mononuclear cells and granulocytes, which are 2 very broad classes of cells. And then you can continue to split your mononuclear cells sequentially over and over again to get even more specialized subtypes of immune cells. So a lot of immunologists do this like on a daily basis, but some of you in the room have maybe have done this before. In practice, it's very arbitrary where you draw these boundaries to delineate a different cell population. It's really tailored towards already predefined combinations of markers that people know to look at. And it kind of, in that way, limits novel discoveries of populations that haven't been well characterized with respect to some outcome of interest. And so we sort of want to automate all of this to not only save time, encourage more reproducibility, but also prime novel discovery, because hopefully we find a cell type that hasn't been well characterized in the context of a particular disease. And so we developed a very scalable multi-sample clustering strategy, which we broadly refer to as meta-clustering. And the input to this algorithm, right, you start with your multiple patient samples, which I told you about in the schematic a few slides ago, right? And by the way, you would definitely have way more than 4. I'm just using 4 to illustrate a small number. And then the first thing that we do is we actually do unsupervised clustering or binning cells into different populations independently in each sample. So each of these points is representing different populations of cells that were found, and broadly, colors are used to encode cells of a particular type. So for example, in the first row, we found 2 types of cells. In the second row, we found 3 types of cells, etc. And then what we do is we extract these cluster centers from each of these individual samples. So, this dark purple versus pink from the first one, and we concatenate these into another big matrix, and then we can cluster this one again and get consensus immune cell types. So these produce, things that we're used to, like T cells, B cells, monocytes, macrophages, assuming you started with PBMCs or whole blood. But really, what comes out is a bunch of consensus cell types that were detected across all of these individual samples. So once we have those cell types, we can engineer 2 types of features to emulate what we would have done with manual analysis. The first is frequency features, where we basically, for every cell population, just count the number of cells that a particular patient has in each of the cell types. So if you have K cell types that you identified, or K clusters, this results in K features. We can also engineer functional features. These are things that sort of are readouts of signaling activities or, transcription factor expression in particular immune cell types. And we define those also for every different cell type. So we have K cell types. This gives us K times F total features. And so in the end, we can make a matrix of patient samples by engineered features where every row is representing a different patient. And every engineered feature in the column is either one of these frequency features or functional features. And then finally, we have a matrix that we can use for machine learning. We can give this to any classifier and classify the rows of this matrix depending on what our question is. So we use this, this is a visualization of the approach, but I'm going to break down sort of what we did with this information. So in this plot, what we've done is we've taken the cell types that were uncovered through this clustering approach, and we projected them into 2D just with PCA. So every point is actually representing an entire cell population. And then we could annotate these cell populations based on the expression of canonical markers. So you can see we found all kinds of stuff: monocytes, Tregs, NK cells. And the particular question that we were interested in here is, how do glucocorticoids or steroids affect the immune system after surgery, and how patients recover from surgery? But I'm mostly using this example to illustrate to you, how we could package this information and extract information from it. So this was a longitudinal data set, and I'm showing you here visualizations for 1 hour, 6 hours, and 24 hours after surgery. And so you can use a map like this. And for example, if we look at classical monocytes here, they're really red. And what we were able to infer about classical monocytes is that their frequency decreased in a significant way after you got if the patient had gotten a glucocorticoid after surgery. And this is not surprising because steroids are very immunosuppressive. So you would sort of expect something like this. But what's really cool is that, this was done basically with no human involved. I mean, we just took the data, applied our algorithm, and this was the result that popped out. But when we actually compare this to what you would get if you do manual gating and manual analysis, we also see at 1 hour that the blue group is representing the steroid or methylprednisolone group. And they also had significantly down, or reduced frequencies in classical monocytes 1 hour after surgery. So this is just one example I wanted to highlight for how you could use an algorithm like this to make agnostic discoveries. So, you know, somebody did manual gating in the end, in the second row here, but like we could have reached similar conclusions just by applying our algorithm. And we can also do this for functional markers. So here I'm showing you I-kappa B. Also in classical monocytes, here I wanted to highlight what's happening at 24 hours. So here we observed that the expression of I-kappa B tended to increase if you received a steroid after surgery. So again, if we look at classical monocytes, we can see that the expression of I-kappa B was actually a lot higher in patients that had received steroid after surgery. Again, so our fully automated analysis recapitulated what you would have seen through manual analysis, which is really nice. So you could use this approach, and now we are using this basically if people have CyTOF data and they want to know what cell types they should focus on or what signaling pathways they should focus on, we can basically do something like this in an hour and give them maps like this. So the most recent place that we've applied this is in studying the effects of antiretrovirals on HIV patients. So this is in collaboration with Nilu, my collaborator, and this is work done by Jolene, and the CyTOF was done by Alexis. These are both graduate students at UNC who actually, no, they're not graduate students. They both recently graduated, so they're no longer. But basically they applied this approach and found all of these major cell types. And in this case, what we were trying to do is actually predict time since having received an antiretroviral therapy. So we did all of this clustering feature engineering business, and then we actually trained a regression model to predict time since having an antiretroviral therapy. So here I'm plotting the model coefficients. So a high model coefficient would mean that you're more highly associated, or like a positive magnitude, high magnitude coefficient would mean you're more positively associated with a later time point, and a bigger magnitude negative coefficient would mean you're more associated with an earlier time point. And by then zooming in on important cell types, such as the change in BCL2 expression in B cells, this gives us, for example, a nice marker or some indicator of a particular function in a particular cell type that changed after having received this therapy. So you can use a model like this and the coefficients that come out to really then get an understanding of what were the important driving cell types and their functional responses that happened after you received this antiretroviral therapy. And so you can see the example that I highlighted here of the change in BCL2 in B cells, this actually did indeed, well, all of these examples are changing longitudinally, but you can definitely see how there's, an increase in this as you have more days post antiretroviral therapy. So I think work like this is nice if you really want to have a mechanistic understanding of exactly what a treatment does and what it's changing, again, either in terms of frequency of cells or in terms of function of cells. And really depending on the question that you're interested in, whether or not frequency or function information is more useful, again, this is sort of application specific. Okay, so moving from this course in descriptive setting, we thought, what if we can completely change the paradigm and not have to do any clustering or arbitrarily defining cell types? What if we move from this course descriptive setting and instead focus on just using a fully deep learning approach to take a bunch of samples that are profiled with a single cell technology like CyTOF, learn a representation of those samples, and then use those for prediction. And so Haidong, who's my student here, did this. And basically we borrowed, when I showed you those little point clouds at the beginning of the talk of airplanes versus chairs, well, I guess those people never realized that someone would steal their method to solve like a non-toy problem, but we basically took their method and adapted it to this problem. And so remember what I was saying is that all of those instances of point clouds are basically samples. You could think about each patient here, like this was the chair group, this is the airplane group. And so first we need to come up with these vector representations, right? We need to learn these. And this is going to be a totally mathematically abstract representation of these data. So we basically need to take all of these cells, do some kind of mathematical operation, and then get a vector out such that when we give that vector to the model, we can accurately predict these patients. And so the sort of way that I want to summarize how this algorithm works is that on an individual sample level, the first thing that we do is we learn a representation for every individual cell. So for example, if we're looking at the green sample, all of these cells would have a different vector representation. Let's say 128 different numbers that represent each cell. And these dimensions are totally abstract. They don't mean anything biologically, so interpretability is very low here. But diagnostically, this is a great tool because it's very accurate in how it predicts things, but it's not good if you want to communicate with someone about something mechanistic. And so this is something to keep in mind, the trade-off between fancy approaches like that use deep learning and more traditional coarse feature engineering approaches. And so what we ultimately do is, once we have all of these samples, you can think about this function as some deep learning architecture. So it has, I don't know, all of these architectures are kind of like a voodoo to me, but we have an architecture that works, that for every individual cell will come up with a vector representation. And this is my favorite part to emphasize here. So what we get out here is a matrix of cells by embedding coordinates, right? And again, this is probably like 128 dimensions by default, right? So this is a particular sample. We have 128 different numbers, right? But I told you we need to actually get to a single vector. And so, this is crazy because if you did this on the raw data, it would never work. If you were thinking about the raw data, looking at the proteins in the cells, you could never just like average the rows of this matrix. So I'm going to draw a few rows of my matrix. You can never just like compress it by averaging the rows, but it turns out you can do a pooling operation. So to do a pooling operation, you basically go by each column, you take that column, all of the cells there, and you have a lot of choices about what you can do to summarize all of that information into one number. So the way that we particularly do this is with max. So let's call this vector x, right? We get this column, which we're calling x. We just look for the max of x, and that's our number that comes out. And then we just go through and we do this. And this is a max pooling operation. And when you do that, right, you get a bunch of different numbers out, right? Let's just say y1, y2, up to y128. And then you can have your nice vector representation. And so, yeah, this is a way that you can finally compress the data into a single vector. Okay, so how does this work in practice? Well, we compared this to a few other deep learning and machine learning based featurization approaches that don't require any gating. So our approach is called CyTOF, which is in green. And in this data set, we were comparing patients who had been exposed to HIV and remained uninfected from those who were unexposed. And, some of these other ones like cell CNN. So this is another approach where, again, you drop the samples in and you learn an arbitrary representation of them. That one is doing pretty well, but we're just a little bit better. And I think the scalability of our method was also a bit better, which is not shown here. And then CyTODX, which was around when I was a postdoc. So my student probably thinks this method is very old. I think it was published in 2018. This is actually just a very simple regression model where you first you concatenate cells across all files and their associated, or all samples. Sorry, I'm talking like a computer scientist. To take the cells across all samples and you concatenate their labels, and then you just train a regression model and then use that. Basically, again, they have some kind of a pooling operation based on the scores that come out for individual cells. But yeah, that one is the only one that doesn't work that well. And we tried this in our paper on a bunch of other data sets, but I'm just showing you one illustrative example here. Okay, but then we wanted to keep going a little bit and make this a little bit more nuanced. And so the original CyTOF formulation basically set up the optimization problem when it learned representations for each cell by minimizing a binary cross entropy loss. So I told you before that f of theta is our deep network. This is like the parameters of this deep learning model that we're trying to learn. Xi is representing a bunch of data from, or like data from a particular patient. And then yi is representing the sample, or the label of sample I. And so what we wanted to do is not only focus on training this model based on an outcome of interest, we also wanted to incorporate other kinds of clinical information. So for example, in this application, which I'm going to explain to you, we were interested in looking at different features of patients after they had been infected with chlamydia. So, notably, there's this phenomenon called ascension, where the virus will ascend your reproductive system, and this is really bad because it leads to infertility. But about these women that came into the clinic with chlamydia, we also had a lot of other different kinds of clinical features about them, like how long they've been on contraception, their STI history, social information about them. And so we wanted to be able to incorporate such information into our approach. And so what we did is we came up with a way to add a term to our loss function. So basically to modify our loss function a little bit in a way that takes into account this information. So what we do here is if we have a target patient of interest I, we get two different pairs. So we get a patient J, which is different in terms of the covariates. Again, covariates are just extra information about the patient. And we get a patient K that's similar in terms of covariates. And then we basically look at figuring out how do we enforce that patients that are known to be similar in covariates end up with ultimately similar vector representations than patients who are not similar in terms of the covariates. So what we do there is just tack on a penalty term so that if the difference, if the patients were more similar in patients that were different in terms of the covariates, then this loss function would not be as good. It would basically add a penalty. So we don't want that. And then, here we're predicting ascension status in these women. And you could see that when we compare it to the original CyTOF model, which is in blue, you could already predict it well, but when you incorporated this extra information about these other covariates, you can predict this even better, which is really nice. So this is just one example of how we can tie in this extra information. Okay, but doing all of these deep learning things, right? We have this practical problem in that we have too many cells. So we can't fit all of those cells in the GPU. So we need to, unfortunately, we can't use all of the cells. We have to throw some away. So there's a bunch of algorithms which are called sketching algorithms, which basically do what it sounds like and reduce the size of the data set or find a limited number of cells per profiled sample that will effectively still encode all of the information about the entire immunological landscape, right? So you can imagine that there's different frequency patterns that can arise in the single cell data set, right? You could have very common cell types like is shown in orange, or you could have more rare cell types like is shown in blue. And then, existing sketching approaches will either just draw a random sample and just randomly pick a subset of cells, or it will do something more sophisticated. In this case, geometric sketching is an algorithm that was developed in 2019 that will just try to sort of enrich for cells that were more rare in the data. So it tries to find cells that it tries to like oversample these rare cell types so that they're not missed in the smaller version of the data. But if you work with immunology data, you know that the frequencies of cell types are actually very important for prediction. So we don't really want to be there to be artificial oversampling of rare cell types. In fact, when we downsample the data, we want to make sure that the frequencies of major cell types actually can be preserved. So we wanted to develop a new sketching approach that would preserve these relative frequencies, but still include rare cell types, with their respective frequencies. And so we developed this approach, which we call distribution-focused sketching, because the idea is that it preserves a lot of the underlying statistical properties of the original data, but most notably, it preserves frequencies across major cell types. So when we were ultimately evaluating whether or not a subsample of the data worked well, we were really looking for, did it preserve frequencies, and did it preserve more theoretical properties like singular values that were in the original data? Did the distribution in the sketched version of the data look similar to what you would have had in the original data? And then finally, for practical use, could we use the sketch version of the data or a much smaller version of the data, and could we still accurately predict our outcomes of interest? Okay, and so we have this iterative algorithm where we start with an empty sketch, right? We don't have any cells, but what we're trying to do is to find a limited subset of the entire single cell data set. And so sequentially, right, we're trying to generate a sketch of size M, so M sampled cells. And I don't want to go into all of this, but it's not too complicated overall. But every time we add a cell, the question that we ask is the cell that we add maximally different from any of the cells that have already been added to the sketch? And we evaluate this through some kind of a kernel function like an RBF kernel. And so the cool thing about this is that these properties that I mentioned at the beginning, if I would have needed M cells to encode properties from the original data set succinctly, our approach shows that we actually only need square root of M cells to achieve those properties. So that's really nice because square root of M and still retaining all the properties is really much better. And, we showed that this works. I told you we wanted to preserve singular values, and we compare our approach, which is the kernel herding approach, in orange. So a lower value of an L1 distance between the singular values in the original data set and the singular values in the sketch. As a function of sketch cells, you can see that this difference decreases, right, as your sketch gets bigger, which makes sense because you're converging to the real data set. But we really do better than these other approaches. And then in particular, what I told you we really care about, right, is preserving the frequencies of major cell types. So we looked at taking our sketches and binning them into different numbers of clusters, so 10, 30 to 50. And then again, we looked at the frequencies of the original data set across these clusters and in the sketched version. And you can see that we also do well in orange at preserving the frequencies the best, as indicated by lower L1 distances here. So this is really nice. Oh, and this I forgot to mention was on actually a single cell RNA-seq data set where we have patients with multiple sclerosis and control patients. Okay, so we solved one problem, but then we quickly encountered another one when we moved to imaging mass cytometry. So, I mentioned this briefly at the beginning, but in imaging mass cytometry, we want to not only measure proteins in individual cells, but we want to take a picture of the tissue which those cells come from, and then stain them according to all these different markers. And then we want to be able to infer so-called cellular microenvironments or prototypical combinations of cells that tend to co-occur in patients that are of the same type. And all of this work has been really developed for, I would say classical immune cells, right? And didn't really consider other complex problems that arise in brain tissue, for example, or spinal cord tissue. So this is all well and good, right? There's algorithms that work well for this. This is from patients with tuberculosis granulomas, and these result in nice spherical immune cells that we can easily find in the image. But unfortunately, when we move to something like this is our data from a patient that has ALS. And, we care about a lot of different properties in this image. Like we care about neurons, we care about microglia, and we care about pathologies of interest or like protein aggregates. And so when we apply the out-of-the-box segmentation approaches to find these different cell types in the data, we quickly encountered a problem because we could not actually segment this entire neuron. So this is probably appearing a bit blurry with the light, but this neuron has this nucleus and this process that comes out of it. None of these segmentation algorithms actually get this process, they only get this nucleus. So, this is one thing that we're interested in working on improving, right, is this mesmer approach, which is sort of the state of the art for just regular spherical immune cells. It's been trained on lots and lots of data of spherical immune cells, but we're trying to basically adapt these approaches or even develop approaches that are not even required for you to segment cells. We're trying to basically adapt these so that we can find all of these structures of interest, such as neurons and microglia, which we don't have markers for here, so that we can really study how those are situated with respect to each other, because that actually has a lot of significance in neurodegeneration, for example. Okay, so, I'm going to briefly talk in the last 10 minutes about some of our future work that we're doing now related to neurodegeneration and microglia in particular, but I just wanted to summarize sort of the main algorithmic contributions that I discussed. So we have these different featurization strategies that have trade-offs between interpretability and accuracy, right? The coarse versions are better at being interpretable because we know what cell types they're representing. And the deep learning based approaches have maybe better diagnostic potential. And then we use sketching as a pre-processing tool for more efficient analysis. And finally, the challenge that we're currently working on algorithmically is actually featuring for these IMC data sets where we don't just have spherical immune cells. So all the software that I introduced are the algorithms. We have open source software for all of these, so you can use them freely. And we always like to get feedback about how well our methods are working and how usable. I will say the kernel herding is very easy to use. I think this is our best one in terms of like usability. I also think it's a good paper, but in terms of usability, it's nice to use. Okay, so I wanted to touch briefly on my new main interest, and this is actually a project that I did over the summer, and now I handed it off to some students, so hopefully next time we all meet, I'll have updates about this area. But basically, I've become interested in the interplay between the peripheral immune system and the central immune system, and in particular, immune cells, circulating in the blood versus what's going on in the brain with microglia or resident macrophages of the brain, for example. And so this is a figure from a really nice review article in 2022, which is actually about humans under stress and how this both impacts the peripheral immune system, and specifically monocytes in the peripheral immune system, and how this infects microglia, and how once your monocytes get a bit perturbed, there's basically crosstalk with the microglia, which wreaks havoc in general in your body. So this neuroimmune focus is effectively aimed to study the interplay between these two very complex systems. And so, yeah, microglia are basically these really powerful little immune cells. And when you're aging or you're experiencing neurodegeneration or in general psychiatric disorders, microglia will really respond in a lot of different ways in terms of their inflammatory profiles, or if they're producing cytokines, and even their morphology changes. So then this changes how they can interact with neurons, which ultimately impacts brain function. So, they weren't really viewed as doing anything useful for a long time. And then I would say within the past 10 years, people finally realized, wow, microglia are like major players in a lot of diseases. And now they're very, very popular and widely studied. So what I did over the summer is I basically took a single cell RNA-seq data set of a bunch of different mice that were profiled. Well, they were post-mortem brain samples taken from mice at a bunch of different ages in their life. So from embryonic stage up to old age. So each of these dots here is representing a different mouse. And then I basically used the feature engineering approaches that I told you at the beginning of this talk. And I took those vector representations, one for each mouse, and projected them into 2D. And, you can see that the mice separate really well by age, right? You have your younger mice over here, like this P5, and then you have your older mice sort of clustered here. And this is just based on frequencies of particular immune cells. And when I took frequencies and I gave this to just a random forest classifier, I could, on average, I did this 30 different times with different train test splits of the data. On average, I get an accuracy of 0.8 in terms of predicting which age the mouse was. And I also did this by looking at inflammatory features or expression of particular inflammatory genes in different subsets of microglia. So I think I forgot to mention this, but basically we enumerated a bunch of subsets of only microglia, and we can accurately predict mice age. And then when we augment these two sources of information, we don't really do much better, but this is what we're working on now, is how do we pick features from each one of these modalities and combine them in a way so that we actually can improve prediction. And so what was driving these? Well, there's actually a lot of characteristic changes. If you look at feature importances that come out from the random forest model, some of them are very expected, like ApoE is a very common microglia marker. But for example, in subtype 6, you can see, which I haven't given you any information about what these subtypes are. I'm just telling you there are different subtypes that have certain interesting features that can drive these predictions. CCL6, for example, was coming up as being expressed in early life, and then the expression wears off in subtype 9. CD14, not expressed a lot in early life, but comes up later. And then we could say the same for frequencies. So this is sort of like a direct application of all the algorithms that I just told you at the beginning, which were actually developed for CyTOF, but actually work well for this problem as well. Okay, And, I'll just quickly say that, we're not only doing this on one data set, but we're actually trying to find subtypes across a bunch of different single cell RNA-seq data sets on either aging, neurodegeneration, or some kind of dynamic psychiatric disorders where patients change over time. We're trying to find salient, or what we're calling an ontogeny of salient microglia subtypes that are associated with dynamic progression, whether that be age, whether that be neurodegeneration status, or whether that be progression psychiatrically. And we're doing this with this algorithm that we developed called DELV, which basically is designed to come out or infer, particular gene expression programs that you could use to recapitulate some biological trajectory of interest. And this is Jolene's work, but if you're interested in this, I highly recommend checking out her. Well, sorry, I didn't put the bioRxiv link, but we do have a preprint about this on bioRxiv. Okay, so just to summarize, I know I talked about a lot of different applications and featurization approaches, but I wanted to conclude with saying everything I talked about and more, we have open source software for. So all of this is on my website, but the methods I talked about today were Cytoset, kernel herding, sketching, VOPO, which was our coarse feature engineering approach, and DELV, which is our dynamic trajectory inference feature selection approach. And then we have other stuff which I haven't touched on, notably like multimodal integration for single cell with other omics. We have other cellular summarization methods, and then we have methods to make our graph representations of the data smaller. Okay, so with that, I'd like to thank everybody for your attention. Thanks for meeting with me and spending time with me. I want to thank all of my amazing students. So Haidong, Alec, Chi-Jane, Emma, Yuchen, Sneha, and Lovna. Jolene did a lot of the work in this talk, and now she's a postdoc at Stanford. And then I want to thank my amazing collaborators. I really hit the jackpot with collaborators at UNC, so I'm really lucky for that. And thanks. Happy to take any questions. Sure.
Great talk. Really interesting.
Thank you.
Just a question on the CyTOF method. Is there, like, I think you mentioned that usually about 40 different markers.
40 to 50, yeah.
Okay. Is that like the minimum number of markers, or after that you're stopping to see diversity in cell types in cell profiling?
No, I don't think it's that. I think it's that it can't handle more than 45 to 50 technically, right?
Okay. See more cell types.
I think that you could, well, that's a really interesting question. So it's really important in general when you do these experiments to design panels that are tailored to look for cell types that you're interested in. So if you had chosen very diverse protein markers that were not redundant, I feel like you could find many populations with fewer markers. But I think if you kept adding them, you might find different functional states, or yeah, probably even more nuanced cell types. But I think at some point, if you're just throwing a bunch of random stuff in there, it won't necessarily help.
Okay. They're just attaching to the surface, right? You're not like...
Yeah, they're served. Well, some of them look at intracellular protein expressions, but for phenotyping, these are all surface markers.
Impressive work.
Thank you.
I didn't quite get the transition from CyTOF data to the 14.
So it depends. If we use the cluster-based approach, so this one, right? So first, well, let me start here. So the first thing that we do is we cluster all of the cells from all of the samples into, we denote each individual cell as, like, let's say 1 of 3 cell types, right? So that's similar to manual gating, where instead of gating for 3 populations, we would have just clustered the data into 3 populations. And then once we do that, in every different patient, we can go across each of those populations. Let's say we have K of them. And a feature that we get is like the frequency. How many of that patient's cells were in each population? So that gives us K different features. We can also look at functional markers, let's say expression of I kappa B, STAT3, all of these things that we're interested in. Let's say we have F of those. So we can look at, for all cells within a cluster for a particular patient, what was the mean expression, let's say, of one of those functional markers. So that gives us even more features that we can put. So you can take all that information, put it together, and one row of this matrix is then the information for one patient. Now, what you were asking about the supervised approach, that's this, where we actually use the outcomes of the patients to come up with feature representations. And this is using a training data set that would be predictive of the outcome. So those help to determine the properties of this neural network architecture. That in that case, the supervision does actually play a role into the feature representation of the data.
I really like this. Get accuracy.
Yeah. No, we're not. We can do any number we want. That's just the default setting. It's just sort of in the machine learning world. It's what they use, so it works okay for us. But yeah, this would, like you said, this one for diagnostic purposes, if you had trained it on a bunch of samples that you knew the labels of, a new person comes in, we just throw it into the model and we can get a prediction of, you know, what the phenotype ranking is.
So with your feature extraction using the maximum function,
yeah,
I would suspect that would be highly dependent on any normalization of your samples.
Yeah, so that one is after we get these like mathematical encodings. So I don't know if that's, if you're saying, was the original normalization affecting that? Is that sort of what you're asking?
That's sort of, that's at least part of it.
Okay.
Then also, do you only get a single value that would be max? Or what happens?
Or
can you? I'm just thinking of different ways of doing it and how you potentially. And I'm not sure if one would be any more or less accurate than another. But say you've got a distribution of features, and maybe it's like the top 5%. Really, you can label all those as max.
Yeah, that's a good point. Or even not just coming up with one number per column, but let's say if we, let's say, wanted to look at the top, I don't know, 5%, we take, exactly, per column. Yeah, we haven't explored, the only thing one of my students has done has looked at median pooling instead of max, which she found, and that was for that covariant augmented version. She found that in that case, the median pooling was working better. In this case, max pooling. But I think that's a really interesting question about like what other operations could be done that might improve performance. And about the normalization, so the nice thing about CyTOF is that we don't, I mean, we might correct batch effects, but the only really data transformation that we do is an ArcSign-H transformation.
We're thinking of applying those to SimSAR, for
example. Which are a nightmare.
Very large.
Yeah, that would be, yes. With all the zeros, that would be, I think that wouldn't work. Well,
so the max, that's what got me to the max idea. I thought that was brilliant, is that you could, it could still be used that way, that the scaling of the max value.
Yes. But another thing is that when these embedding coordinates come out, they're not really in that original data space anymore. And yeah, I do want to point this out that, so we did try doing the pooling, I think sort of like you're suggesting, of cells by features, like original, let's say they're proteins, right? If we just do max pooling on this, it's actually nonsense, which I was so happy about. I told my student, if that works, we're in deep trouble, because that's so simple, right? We basically are out of business, but it didn't work. Yes, or any single cell. Oh, okay. So, I wasn't a fan of, well, okay. So I really like CyTOF for a couple of reasons. And I really like Flow for the same reasons. So Flow, actually, you can get tons and tons of cells. Like, it is super high throughput. You can measure less proteins per cell. But I really like the idea of measuring proteins because I feel it's really what's there, right? But and the other thing I like about CyTOF and Flow is that you really know ahead of time what you want to measure in your cells. It's not as much of a fishing expedition. When I started working on RNA-seq, I gained an appreciation for, when I started working on microglia, I gained a appreciation for RNA-seq because I really didn't know what I was looking for at all. So, I think they both have their place. I feel like transcriptomic is maybe a little bit more exploratory. It's more broad, all-encompassing, but I feel like CyTOF is really good for phenotyping immune cells, and in particular, if you know what kind of cells you're looking for and what kind of like signaling responses you might want to look for. I feel like it's just highly targeted. And then the fact that it's a protein expression, and I think the data is a bit easier to work with than single cell RNA-seq because there's not all these filtering steps and mapping. It's like a huge nightmare, RNA-seq. I feel CyTOF, you just like download, or someone gives you the data, you do an ArcSine H transformation, and then you're ready to go.
That's similar to what Cody brought
Mhm.
up, the number of features that you can get.
Yeah.
So,
Yeah,
but it just adds a lot of covariants that aren't relevant to what the question is.
exactly.
So that you can find off. No, I think that means that, you know, do that, do the single cell.
Mm-hmm.
But when you go to the
Mhm.
you know that work to only whatever 8,000, 10,000.
Mm-hmm. That is true. That is true. Uh-huh. That is true. It does, yeah. Yeah, like these microglia data sets, I really had no idea what I was looking for. If someone asked me to design a CyTOF, I mean, there are some papers doing CyTOF on microglia, but they're very canonical microglia markers that aren't really interrogating the functions of them. Oh, you don't. I mean, I guess because they produce cytokines and everything, so they do sort of have inflammatory response. Yeah, yeah. I mean, they're sort of. I guess. I mean, would you prefer I call them glia? Yeah. Yeah, I guess they're calling that the central immune system these days. Why is that? I don't know. They're not spherical, yeah. Well, they're, yeah, they, Yeah, it's a good question. I guess they are, I guess, yeah, what does it mean to call something an immune cell? I think it's, I say broadly, I said this in my third slide or so, immune cells are broadly providing protection, right? And I feel like microglia actually are providing protection. No, yeah, yeah, uh-huh, yeah, good point, yeah.
So that always makes you think that you one set, not actually.
Hmm. Hmm.
I really like it. I think that's what it's like. You really know.
Mm-hmm.
Yeah, we were very not, we were there.
Yeah, I know it's complicated though, and it's practically difficult because you can't get you can't kill people to get their brain.
But it's not controversial, so I'm not going to that. Neurodegeneration is, it goes in fits and stuff.
Mm-hmm. Yeah. Mm-hmm. 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 Thank you.
Well, let's thank her one more time. Usually not on you.